my pleasure to introduce Adam MacLean, who's giving what I think is the first seminar of the year, isn't it? And then we have another one on Wednesday, just to remind you, at the same time. So Adam started out as a mathematical physicist at the University of Edinburgh, did a PhD in systems biology at Imperial College London. He was a postdoc in Oxford and then UC Irvine, and he joined USC in 2019. So he's currently an assistant professor in the Department of Quantitative and Computational Biology that I have a old, long historical connection to. His work is supported by a number of grants, for example, an NSF Career Award and an NIH Mira Award. So he's going to tell us today about inferring cell fate networks that control outcomes in complex tumor-immune microenvironments. Adam, it's a pleasure to have you here. Thank you for coming on this complicated day. And let me give you this device back. Great.
All right. Hopefully you can hear me well on Zoom. Thank you very much for the kind introduction, Simon. It's really a pleasure to be here, and I'm very excited to talk today about some recent work from my lab at USC on inferring these cell fate networks that control outcomes in complex tumor-immune microenvironments. So at a high level, my lab really focuses on the theory of cell fate decision-making. So I'd like to just get us all on the same page and talk briefly about a survey of cell fate, so what it is that a cell can decide to do. So this is my cell, as you can see. The first thing it can do is to do nothing. This is important. It can be an active choice and an essential one. And then my cell can divide. This is, of course, fundamental to all of life. My cell can die. This can also be very much an active choice and essential to multicellular life. And it can migrate or transform from one state into another. Finally, it can differentiate, which I put down here at the bottom. It's not fundamental in the sense that it can be made up of these cell fate decisions above, but I put it here because it's so essential to stem cell biology and how we think about cells differentiating. So I like to keep in mind that the kind of inception of stem cell biology was itself very much theoretical and mathematical from its very earliest foundations. In a few papers in the 1960s, so 60 years ago, these Canadian scientists, Till and McCulloch, they used mathematical models combined with experimental assays where they pulled cells out of the bone marrow and they cultured them, and they found specific cells that were highly proliferative and had the ability to both self-renew and differentiate. In other words, they had these essential hallmarks of a stem cell. And this was years before, 25 years before these stem cells were actually found and quantified in vivo in their niches. But of course, cells do not make these fate decisions alone. And today I'm going to talk about the context of cell fate decision-making in the tumor microenvironments, or TME. So just briefly then, there are myriad sources of milieu of signaling and many things happening in this TME. And let's go through them from roughly speaking, from the smallest scale. We have, of course, genetic variability and tumor evolution over time. We have these dynamics of epigenetic factors that can control the states of cells that way. And then we have transcriptional states, the gene expression within each single cell. Continuing then like up, thinking about cells as a whole, we can think about just as asking what cells do we have in a specific time point in a specific TME, the composition in terms of the cell states or cell types present. And then we can think about higher order effects such as cell-cell interactions and spatial context and spatial organization. So clearly, to some extent, you cannot separate all of these different levels of heterogeneity, but we also have to focus to ask biological questions, and my focus today will be kind of in the middle of this hierarchy from the transcriptional scale to that of how cells interact in the context of different treatments in the TME. So the biggest way I can frame the question of this project is, how do combination therapies improve outcomes during breast to lung metastasis? So I'd like to just introduce the biological system, and my collaborator, Evanthia Roussos-Torres at the USC Keck, asked this question: clinically, can HDAC inhibitors, so histone deacetylase inhibitors, combined with immune checkpoint inhibitors, ICIs, be used to treat metastatic breast cancer? And they recently published this phase 1b clinical trial where it was shown to be a safe combination of therapies. This is entinostat, the HDAC inhibitor, combined with checkpoint inhibitors. And you see, so it's a very small, this is a phase 1 trial, it's a very small cohort, and we see some indication of improved outcomes, especially in the triple negative cohort of patients. So the answer is a tentative yes. But when we look clinically with kind of bulk sequencing, which is as much as can be performed, it's very hard to, or at least as until yet, we have not distinguished anything that might be a clear mechanism for what is driving the effect of these drugs. So that's where we came in, and we turned from the human, where it's a little bit hard to measure things, to the mouse, where we get to measure everything we want. And it's useful to disentangle these mechanisms. And we're going to study breast cancer metastasis in the mouse, specifically using this syngeneic model of spontaneous breast cancer metastasis, where previously my collaborators looked at the primary tumor and the TME, so the different populations present in the breast of the mouse, using this NT2.5 cell line derived syngeneic breast cancer model. And when they looked there at the immune suppression under the effects of this entinostat drug with checkpoint inhibitors, they had a hint that perhaps these STAT3 and NF-kappa B pathways were mediating some of the immunosuppressive effects. But this is challenging because it's in the primary tumor, and we really, the clinical effect we see is in metastatic cancer. So they made a new cell line called NT2.5 LM, which is derived from NT2.5 mammary tumors, and these cells spontaneously metastasize specifically to the lung. So that lets us, in a reproducible way, study the TME in this spontaneous breast to lung metastasis model. And I'm going to get into all this composition below, but just for now, you can see that the survival is worse, which you probably expect with this like spontaneously metastasizing model. And they're also growing at a faster rate. So then when we look at this lung TME, where we're having these metastases grow, and we're going to treat with the 6 different treatments, and basically combinations of E is entinostat, the HDAC inhibitor, and P and C are checkpoint inhibitor therapies. And we see like small, but in the case of EPC, significant differences in the survival of the mice treated with the combination therapy. So the first thing we asked was like, what's going on in the tumor cells with treatment? And let's look closely there. And the answer is like very little, in a word or two. The tumor heterogeneity is low, and the treatment effects are subtle. And maybe that's not surprising. This is a cell line derived model. You know, we look very hard at heterogeneity by gene expression. Can we subcluster populations? And the answer is basically no. There's very little evidence. Maybe there's very small differences that are mostly metabolic driven. So now looking at treatment effects, we use this like linear discriminant analysis just to look at the difference pairwise between vehicle and each of these 3 treatments, entinostat alone, checkpoint inhibitors alone, or the combination. And we see that actually with either PC or EPC, there's like a high overlap. There's very, very little in terms of what's difference in terms of like the entire transcriptional state of these tumor cells. There's a more of a difference. There's a larger difference treated with entinostat alone. But overall, things are quite subtle. That's also the case when we look at differential expression. there's not very clear marker genes or strong changes in the tumor cells with these drugs. So this kind of got us to ask a question of how can we go about quantifying and characterizing these cell state shifts when they do seem to be very subtle. And a PhD student in my lab, Yingtong Liu, developed a small statistical tool for this purpose via iterative logistic regression for classification and marker gene identification. And so the idea here is really simple. We're going to kind of pre-filter some genes and choose a subset of genes. And then we're going to use logistic regression over multiple rounds where we keep rank genes by coefficient and keep like 80% each time to try to accurately quantify the differences between two outcomes while retaining only a relatively small set of genes that can be used for distinguishing. So we're going to use like a Pareto front optimization to choose a gene set for this purpose. So as we like reduce the number of genes by iteration, throwing out 20% of the genes each time, like eventually you can see that actually there's some stage you're overfitting and you've got noise, and you have some sort of maximum accuracy in the classification task, and eventually it's going to drop off if you just have not enough genes in your data set or gene set remaining. So we use this like penalized Pareto front, where the red line is like the actual Pareto front. This is the optimal balance of accuracy in classification and number of correct genes. We're going to use a penalized version where we can just force it to be fewer genes. So that lets the accuracy drop, but hopefully not by too much. So we're still doing a good job. So just having a look at how this works, a couple of examples one on a synthetic data set and then one on a large non-cancer neuronal data set, just for validation. So this is Splatter, allows you to simulate gene expression data with different like DE scale factors. So there's like noisy genes which are not differentially expressed, and there's truly differentially expressed genes where if the scale factor is high, then the average fold change is high, and this is really easy. And in this case, like kind of anything, everyone can do a good job. Like you can completely accurately classify the two groups. As the scale factor gets low, it gets hard. And basically, we're showing that this ILR, our iterative logistic regression, is doing a decent job at classifying cells accurately, even all the way down to the kind of like smallest scale factor. At the very small size, we need like a larger number of cells. We can do a good job on 2,000 cells, and we do less well when there's only 500 cells. But here, the number of genes we're using is really important, because there's over 200. Well, there's a total of 1,200 genes in this data set, around 200 true DE genes. So everything else, like LR or these other like tools, the random forest, etc., are using all the genes. And we're using like this number, so somewhere between up to 200 or down to just like 10 or 20 genes. So with a very small subset that we can hopefully have more confidence in, we're managing to pull out the differences between these groups, in some cases even when they're really subtle and quite mixed together. And this is just showing you that we're also getting the kind of, as the penalty increases, the gene set decreases then in size, then we're learning the gene set precision goes to 1, so we're only keeping, in this case, the true DE genes. So just applied to a neuronal data set here, there's lots of different cell types, which I'm not going to go into. We applied ILR to this data set to have a look, and the light colors here are the accuracy on the top, the number of genes in the gene set on the bottom, and with no penalty applied. And so you see we do a pretty good job over this like range of neuronal cell types. And the dark color is when we apply a penalty of 1. So you see a drop in the accuracy of, say, 5%. So you're doing worse, but not too much worse. And then the drop in the terms of the number of genes that you're using to identify this population is really dramatic. This is a log scale. So you're going from like hundreds of genes to, again, somewhere between like 10 and 50 in most cases. So I think this is a nice way to just look at kind of rank marker genes in terms of small importance sets. And this is just kind of looking at the biology of the genes in our marker gene set that we identified, which there was a lot of overlap with this. Sorry, I didn't tell you, the two groups here are healthy individuals and ASD, so autism individuals. So when we get the genes in our gene sets overlap a lot with kind of the known neurological mechanics of autism. Okay, so then going back to this metastasis model in the lung TME, we're looking at the difference in tumor cells in our kind of like ILR gene set, looking at the difference between vehicle and the combination therapy. And just as one example, my collaborators are really interested in this PYCR2 gene, which interacts with the MYC pathway and seems to be kind of differentially expressed. Well, I mean, it is differentially expressed. This is a differential expression, but you can see like it's significant, but like hundreds of things are significant. Like the fold change is really not very big. So in this kind of typical global differential expression analysis, you're unlikely to ever focus on this one. It's like ranked 59 just by like whatever. I think this is Wilcoxon p-value. So this is not going to be in your top 50 things. But when we did this iterative logistic regression, it was in this predicted to be very important gene set. And so that's a prediction that our collaborators are following up on. Now, I should say, please do feel free to ask questions as I go along if you like. If there are no questions for the minute, then let's return to the lung TME and look at our different populations. Now, this is the high-level picture. There are 8 cell types. So basically immune populations, lipofibroblasts, lung endothelial cells, and the tumor. When we look at the shifts in composition with these different treatments, we don't see any significant changes. Of course, well, not of course, we could have stopped here, but my immunology collaborators would not let me stop here when it comes to cell type identification. We had to go much, much deeper, apparently. So this is where we ended up. This is like a mature myeloid cluster. We ended up with the 12-ish subtypes of macrophage, neutrophil, and dendritic cell. Inside the T cells, there were, again, somewhere between 10 and 15 subtypes. And when we started, we did this for every cluster, and we ended up with 39 cell types in total, unfortunately. But that's where we were. And then when we look at the changes with treatment, we do start to see significant shifts. We're using a propeller here rather than like kind of t-test statistics, which is a nice parametric model for looking at cell composition changes. And we see, for example, Th2 cells and T regulatory cells are changing dramatically with entinostat treatment. We also see changes in certain subsets of macrophage and dendritic cell. Okay, so this starts to give us some clues about differential infiltration of these cell types. But clearly, this is not going to explain the whole picture. Now, before I continue with the whole picture, which is basically going to be asking what cell-cell interactions are the most pertinent and could be mediating the effects of these treatments in the TME. But before we get there, I'd like to digress one more time to talk about this is actually the largest cluster of any at the high level, 40% of all cells across all treatments. And these are MDSCs, or myeloid-derived suppressor cells. And again, approximately 2 years ago, when we began this project, I had never heard of this cell type. And yet there were lots of them. So what are they and what are they doing? So I hadn't heard of them. So you go to PubMed, right? MDSC. This is like how it looks. So apparently there's a lot of people that have heard of them and they're publishing papers on them, like hundreds of papers a year for the last decade. So what are they? They're immature myeloid cells with potent immunosuppressive activity. So they're suppressing functions of the immune system. They've been implicated in the regulation of immune responses in many biological contexts, not only cancer, but inflammation, wound healing, autoimmune diseases, etc. They can be split into two subtypes based on kind of monocytic and granulocytic or polymononuclear MDSCs. This is really complicated and contentious, and I don't want to get into a neutrophil fight because those get nasty, and I'm Scottish. So unfortunately for our purposes here, we don't need to that's not true, I shouldn't say that. But so we're going to be careful about how we subtype MDSCs. But functionally, we don't necessarily know what the difference is. That's all. So I did a second search query this time. Hey, has anyone looked at like simulating the dynamics of these things in the context of the TME? And then do MDSC mathematical model. I can't plot this on here because you wouldn't see it. Like 1-1. Okay, this is a couple years out of date. We started this project 2 years ago. Things have gotten a little bit better, perhaps by our work and others, but it's still like you can't see those down there. So we saw this an opportunity and asked, let's develop a simple model of the dynamics of these MDSCs in the context of the TME and how they might be modulating immune responses and tumor outcomes. How do MDSCs interact with the tumor and other immune populations to influence outcomes? So that was the question that a postdoc in my lab, Jesse Kreger, set out to answer with a simple mathematical model. So we set out to model the tumor immune dynamics. That's my tumor cell. And basically, with apologies to immunologists, I still struggle sometimes with the terminology. I still need to simplify things conceptually for myself. So I think the immune system is doing exactly two things. It attacks invaders, can be tumor cells or other pathogens, and then other populations stop the attack, or potentially remember who did the attacking. And that's it. And of course, then you have to ask who, and then you get into all the complicated stuff in terms of what these subpopulations are. But for me, those are really the essential dynamics to think about is attacking the invader and stopping the attack, the latter being the immunosuppression. Now, of course, the tumor cells interact, they activate these cells, these type 1 cells, if you will, which attack it. And then this is the worst arrow. This is the way in which cancer co-opts the natural immune response of shutting down the attack for its own good, right? To allow metastases to grow and spread. And that's what we were seeking to modulate. So in the context of our system now, focusing based on the single cell data, asking which populations are present at the site of the tumor. Well, these kind of like anti-tumor populations doing the attacking are basically, we see NK cells and we see CD8 cytotoxic T cells, CTLs. And the dominant, the pro-tumor population that's shutting down the attack was the MDSCs. So we're going to develop a simple model of these 4 populations in the context of the lung TME. And I'm going to show you the model. It's a set of 4 differential equations. I've written them out in words. This is basically kind of the simplest terms that we could think of to describe the interactions between these 4 species, their growth, their death, and interaction dynamics. And then we specified this model in full by, we want to model the kind of noisiness of these dynamics. So we used the stochastic differential equations. And we also modeled a delay because we understood from many discussions with our collaborators and from papers that this MDSCs are not initially present at the site of new metastases, but they are recruited and activated at that site. They kick into gear at some point. So we thought it was important to include this delay tau in the dynamics of the MDSCs. So the full model is specified by a set of 4 coupled stochastic delay differential equations, where we use multiplicative noise for each species. And of course, we can like reduce that to simpler models. If we turn off the noise, we have a DDE, a delay differential equation model, and we have an ODE model if we have note a delay in MDSC recruitment. Okay, so we can, play with this model. We can solve it for steady states just to do some sanity checks and see what it does. And it permits two steady states, 2 fixed points. One is a tumor-free steady state. Of course, that's where we'd like to get to. That's typically not where we are. And then it has a steady state where the tumor is at its carrying capacity, and we can look at, in terms of the parameters, what are the different immune populations. But really, we're going to get most insight and progress here, I think, through simulation. So to simulate this model, we need parameters, and we need initial conditions. The initial conditions we take from the data, like the vehicle condition, just like who's who, what cell types are present in what proportions in the lung TME. The parameters that we use are here. It's a fairly large model. I guess, at least in my field, everyone's large is different depending on your, model, system, background, many things. But so this is about 15 parameters. Some of them we know or have some sort of good idea for, and some of them we really don't, specifically like the MDSC dynamics, which has really been looked at in very little detail previously. So in this case, we kind of at least varied things over a few orders of magnitude to try to get an understanding of the range of possible behaviors of these interactions. So let me show you just some basic simulations of the model. First, when we have no delay, so this is no delay and no noise, the deterministic model, that we just have the ODE, and we see that this activation, there's like a biphasic growth in the tumor. Oops. There's a biphasic growth in the tumor, where when there's enough MDSCs, then it's allowed to grow to its carrying capacity. When we add the delay, and we basically get the same thing, but it's just there is a delay. Surprisingly, in the tumor's establishment, I'm using like a log scale here. So these are growing to quite large sizes over the period of 1 year. And then we add noise to the model, and we still see kind of similar dynamics overall. These are just individual realizations, but over many realizations, we see these tumors still grow, and they grow larger when the delay is smaller. When the MDSCs are recruited earlier, but overall, the phenotypes are similar. Now, of course, in the presence of noise, there's a probability that the tumor will not survive. I think I didn't mention that the initial condition for this metastatic, we're looking at metastatic seeding. So it's a brand new tumor. We're simulating this population from 2 tumor cells. So it's very small. And so at some point, these tumors, of course, there's a probability that they will not survive, that they will die out in the stochastic model. That lets us ask just the simple question of what is the probability of establishing a new metastasis from this initial seeding of two metastatic cells and just watching a tumor grow? And how does that depend on the parameters? And it turned out, so this is like our first indication that maybe these MDSC dynamics are important, are impacting the system in important ways. The MDSC delay rate was the most important parameter in terms of controlling the establishment probability and the size of a new metastasis. So here I'm just showing you for different initial levels of circulating MDSCs, then there's always a strong dependency on the size of the delay in terms of the size of the new MET and the probability that it will persist over time. So basically, if your delay is high enough, then you can really you have a much better chance of not allowing a new metastatic growth to establish because those MCs are not recruited early enough to start immune suppression. Okay, so that was a first indication of the model dynamics, but we wanted to go dig a little bit deeper and look at how important the MDSC dynamics are in determining specific tumor response outcomes in this TME. And for that, we're going to do parameter inference on clinical data describing metastases in growth or decline. So here we're using RECIST data to quantify the size of these tumors over time, where these RECIST criteria give you a X and a Y, they give you an area basically, and we just do some very simple math to convert that to a volume. And we're going to look just in terms of relative change. We'll normalize everything, and we're looking at relative change to baseline. And I'm showing you 6 representative tumors. This is a previously published data, not from our cohort, just showing you that some tumors grow over time, some say more or less steady, and then some will decline. And so we're going to fit each individual tumor to the model to ask what parameters control the difference in outcomes. We're not going to fit like a 15-dimensional parameter space. We chose 3 parameters to vary, and we fixed the rest. And we based this on kind of the importance ranked by like the global sensitivity of these parameters. And we looked at, and we also want to choose like different interactions to make sure that we're allowing all the cell populations to vary. So we chose the tumor growth rate, and the rate of like activation of CD8 T cells, as well as the rate of inhibition of NK cells by MDSC. So this is kind of like the immune suppression. That's parameters to vary. And then we're going to do kind of Bayesian parameter inference in Julia and Turing.jl. Not without the stochastic model, we're going to fit the DDE model with prior over these 3 parameters. And we're also going to fit the sigma, the noise of the realizations of the simulation of the DDE. So we did that for these 6 tumors, which either grow or shrink over time. Here I'm just showing you 2. These are like representative. We got reasonably good fits, but clearly not that fitting all of the dynamics. Although these are very noisy measurements. There's a interesting question about what is the appropriate variation, which we're not really tackling here. But we got reasonably good fits for these tumors that either grow or shrink. And when you look at the posterior distribution for the 3 different, so there's 3 parameters. There's 3 kind of 2-dimensional joint density distribution plots. You can see that these are completely mixed up. And this one looks really interesting. We've got this nice rainbow. In other words, the tumors are distinguishable by these 2 parameters alone. Alpha 1 is the tumor growth rate, and the beta 3 is the immune suppression. We're actually surprised by how separable this was, right? So we actually get like this rainbow from the tumor that declines the most to the tumor that grows the most, just based on this joint density plot. So to look at that just a little bit further, we fit a small classification model, a random forest, on the posterior parameters, just trying to predict, will this tumor grow or shrink based on the posterior parameters. And so we do that. We use either like 1 parameter, like of the 3 possible, or combinations of 2 or 3 parameters, and just say with what accuracy can you predict the answer, as in will this tumor grow or shrink. So what you see is when you use the tumor growth rate, right? This is the tumor growth rate, right? And you're asking, will the tumor grow or shrink? You might think that should be easy, but it's like 63% accuracy at predicting that. This parameter alone, the immune suppression, it does better. And then when you combine the 2, you get the best out of any combination of these parameters. You can predict with roughly 82% accuracy, will this tumor grow or shrink? So it told us a couple of things. It told us that we should probably pay attention to the dynamics of immune suppression in this context, and that this specific interaction, this inhibition of NK cells by MDSCs, seems to be really important in the lung TME in the context of a nascent metastatic growth. Okay, so then we've seen that MDSCs are predicted to play an important role in this TME. Yes, please.
So how play on to the delay that you see in MDSC? So the 2 parameters that you identified, the tumor growth and immune suppression.
Yeah.
So what is the delay? How did you incorporate that delay into this context?
Yeah, so we didn't infer the delay rate, so we fixed it to be a constant. So like we saw like that it is important, but like for various for different levels of circulating MDSCs and for various delays, like you see like quite an important control of the eventual tumor size. So yes, in this case, like out of just not wanting to constrain the parameter space, then we just we fixed the delay. I can't remember. I think probably here about 10 days, or maybe 50. Yeah, yeah.
And so that is the fixed parameter and all the other parameters. Okay. But so if that is going to see emergence of DSC, and that's what the beta parameter, right?
Yeah. Any other questions?
Did you hear?
Is there a question online?
Yeah, so I wonder, how did you actually fit the differential equation to the data?
How did I fit the differential equation to the data?
Yeah.
This question. So I did that using, parameter inference, MCMC. So I choose three parameters and prior distributions for those parameters. And then I sample from these prior distributions. And I define a likelihood function, which is here, which is basically just simulate the differential equation, DDE model, and then sample the simulations according to some noise parameter. And then I do MCMC with this likelihood function.
Well, all right. Thank you so much.
Thank you. So the interaction parameter is not where the delay is on the basically growth rate. So recruitment and activation of active MDSCs. And then there's a separate term, which is the interaction between MDSCs and the tumor. Oh, well, MDSCs and this immune suppression, right? So it's interactions between MDSCs are inhibiting the other immune populations, the NK cells and the T cells. And we saw this, that the rate at which they inhibit the NK cells, which is this beta 3, is really crucial in controlling the dynamics and outcomes in the context of this simple model. Now we're going to return to the big picture, the single cell where we have many cells doing many things. And we're really interested in we think MDSCs are important, but we can't just go based on our whims and what we think looks cool, but we need to ask questions of the data to find out what interactions may be driving these treatment effects we see in the lung TME. What cell-cell interactions are most important, and of course, what pathways are mediating those interactions. So, well, just very briefly then, this is probably maybe obvious to some, but I like to stress the point because I think it's underappreciated. Okay, so we have single cell RNA, we have tens of thousands of cells, sure, and you measure all the genes. In our data set specifically, we have 39 cell types across 8 major clusters, and we've got 6 treatment arms. Okay, so that's 39 by 6, that's 234 cell states. If we look at cell transitions, just considering the relevant ones, that's about 700. And now we care about networks. We want to know who's interacting with whom. So if we look at just 2-state networks, 39 choose 2 is 741. 2 states is probably not enough. 39 choose 3 is about 9,000 networks. And that's before you've thought about transitions or with treatment, nor even signaling pathways mediating those treatment effects, right? So I guess it was after a couple of years of really struggling through these data that I reassured myself a little bit with this note that the necessity of the kind of systems biology approaches is, I think, absolute. So how do we go about this, identifying important cell-cell interactions? So we're going to use prediction of cell-cell communication via ligand receptor data in the single-cell RNA. And we do that at the top level between these 8 major cell types. Okay, and we see that the signaling is driven by mostly tumor, myeloid, and lipofibroblast interactions. Now, we want to do this among all the cell types, but 39 is too many. So we constructed 3 separate comparisons of these cell states based on known biology. The first one, like CC suppression, is looking at the interaction between the tumor, the T cell, and these suppressive immune populations. We're then looking at interactions specifically based on antigen presentation. So we're just pulling out many T cell and dendritic cell, the kind of myeloid subsets, to ask how they're communicating. And finally, we look at the interactions that we defined previously in the CIR model that I just presented on. And we're going to look for each of these, and we're going to look at what the most important interactions are. But still, we have a hairball again. It's not as bad as it could have been if it were of size 39, but it's still bad. So what next? Here, there's a really nice paper a couple of years ago that had this idea of like looking for network motifs in the cell signaling data. Basically, the idea is you look over all the possible network motifs, like unidirectional, bidirectional, signaling interactions. You can have self or not self-regulation. And for each one of these, you can look at all the instances of that in the true network. And then you can just permute the network. So look at kind of like graph sub-isomorphisms to get a z-score of, is this motif overrepresented based on what I would predict by chance? So I'm showing you these z-scores. You have to choose like a size, the kind of 2-state motif or a 3-state motif. And you look over all the instances and you ask which is overrepresented, which is a significant z-score. So like for this example, you've got like a fully connected network in the 2-cell case, and so on. So then based on that, we can say these are the most important network motifs of communicating cell types. Then, of course, you have to ask which cell types communicate using that motif. So you can rank the instances of the motif and then study how do those motifs or those cell circuits change with treatment and what pathways or ligand receptor pairs are driving those changes. So that's what we're going to do. So here for the first comparison, CC suppression, the most overrepresented or most significant network motifs were basically these, like in both the 2-state and the 3-state model, that fully connected. So like feedback from all 2 or all 3 cell states, along with this like self-activation or inhibition regulation. When we look at the antigen cell comparison, the dendritic cells, it's quite a different picture. We have this like kind of small motif just with unidirectional signaling onto one specific cell type. And then for the third comparison, the CC model, it's this kind of an intermediate motif where we have a feedback, we have like a feed-forward feedback type of structure here where these 2 populations are interacting and they're both receiving signals from a tertiary population. Okay, so we have these 3 types of motifs that are overrepresented in the data. We have to go and ask which cell types are signaling by each of these motifs. So I'm just showing you this one example from CC suppression now. This is ranked by like total interaction strength. You can pull out the kind of like top networks by strength and ask how does each of these change with treatment. Like you can see specific interactions turning up or turning off or down with these different treatments. And then eventually, of course, we also want to know who's mediating the signals. So this is one of the top motifs that we landed on through this kind of like unbiased analysis of these motifs or cell circuits. So it consists of the effector CD8 T cells and 2 myeloid populations, the metabolically activated macrophages and these subtype G MDSCs. So I'm showing you first like the vehicle and then how it changes with the HDAC inhibitor alone, immune checkpoint alone, and in combination. You can see, for example, when you look at signaling to the effector CD8s, you see that it's down with either from the MDSC, so it's significantly down compared to V with either E or EPC. For each of these 3 interactions that we've chosen, so signaling to CD8s by MDSCs or metabolically activated macrophages, and also signaling to the macrophages from the MDSCs. We look at the pathways that are mediating those interactions. And the consensus across these are all the ones that change with treatment. The consensus across this is 3 pathways. So it's chemokine, galectin, and this integrin signaling via ICAM1. So basically, each of these, I think we think, my collaborators think we're sufficiently confident in to want to test to see, can we replicate the effects of these treatments by cell-cell interactions mediated by one of these 3 things. So we kind of think this is very much where we're at. This is very present and ongoing work, but we have some hints. So I'm going to show you ICAM1. And so here we're doing a T-cell proliferation assay. We pulled out T cells and G MDSCs or macrophages from these Mets lung TME, and then we mix them together in different proportions, and the readout is the proliferation of the T cells. So here, for example, you see the immune suppression when you mix T cells with G MDSCs, that they do suppress, the proliferation goes down. But here we didn't see an effect with when we block ICAM1. So we didn't see an improvement in this T cell proliferation when we block ICAM1. We did see a significant difference here when we looked at blocking ICAM1 when mixing macrophages with T cells. Then we do indeed see an increase in T cell proliferation, but then it's a little bit more complicated because it's more like T cell activation than a reduction of immune suppression, because the T cell proliferation is not actually down just with MACs alone. So then we've got some evidence that yes, like this ligand receptor interaction via ICAM1 perhaps is playing a role in mediating some of the effects of the HDAC inhibitor that we're using in this combination therapy. But clearly that's not the whole picture. There's more going on. And as I say, the kind of individual perturbations for these proliferation assays are ongoing, but what's really hard to do is combinatorial ones, combining multiple inhibitors, because it's just like even one of these experiments, as Clara tells me many times, they're really a real slog to get through in a week, one of these proliferation assays, isolating all the cells from the primary tumor. So we do what we do as theoretical biologists. We go back to the model and try to model the effects of these multiple pathway hits at the same time. So we've got a new initial conditions now. We're changing the initial conditions to be not like a new growing tumor, but the steady state, the amount of tumor we saw at the start of treatment from these mouse models. And we've got all these predictions of the change in the different parameters based on these interactions and ligand receptor analysis. So we're going to model those specifically. We've got, for example, CD8 T cell activation increases 1.4-fold. So we've got a parameter for that in the model. We also have MDSC suppression on the NK cells in this case, reduces to about 40%. So we're going to change those parameters either alone or in combination and just simulate. Now we're starting at the steady state. So the solid line is the baseline and the dashed lines are after the treatment. So we do one separate or both of these parameter perturbations together. And this is the mean field, the deterministic model. This is the stochastic dynamics. And so the red is the tumor. And this is just over 60 days. Basically, you see the probability of extinction is about 50-50. So about half of these tumors will not survive, where if we had not perturbed the parameters, like 100% would survive. And you have to do both. So if we perturb only one parameter, either alpha-6 alone, the probability of wiping out this tumor over 60 days is 0, and the suppression parameter alone is like 4%. So you need both. And when you do both, you can get a really strong effect. If we continue this simulation out over a period of 1 year, then like 84% of these tumors would be, in the mathematical model, would be eradicated. So this is kind of confirming what we see from suppression assays and proliferation assays when we pick out these molecular targets, is that they might well be part of the answer. We think they are, but they're not all. And we are going to need to consider the combinatorial effects of multiple pathways to recapitulate what these drugs are doing in the TME. Okay, with that, I'd like to summarize. I've talked about cell state transitions and inferring interaction networks from single cell sequencing data, and how this analysis of interactions and transitions quickly becomes intractable without considering the big picture in systems biology approach. We have a tool via iterative logistic regression to identify marker genes, specifically of subtle cell state shifts. And I talked about how via cell circuits analysis, we revealed some new mechanisms by which this entinostat HDAC inhibitor treatment is reducing the immune suppression on CD8 T cells via, in part, ICAM1 and adhesive integrin signaling. And finally, integrating this single cell sequencing data with mathematical models, it enables prediction of things that are very hard or impossible to test, such as tumor outcomes under multiple combinatorial treatments or pathway perturbations. This is a very exciting and active collaboration. We've got a lot more to do, and some immediate future next steps include looking at these ILR biomarkers in our clinical cohort to see if any of those can be recapitulated, and also to fit our model to individual patient dynamics, which we have from the phase 1 clinical trial data. With that, I'd like to stop and thank everybody who contributed to this work from my lab, postdoc Jesse Kreger and PhD student Ying-Tong Liu, from Evanthea Rusas-Torres' lab, which has been a fantastic collaboration and very grateful to be a part of, and PhD student Edgar Gonzalez has led the TME work. And with that, I'll stop, and I'm happy to take any questions. Sorry.
You have to read out the questions.
Yes.
Or say them again.
I mean, sure, I have the power. I will do my best.
Any
Sorry.
example questions? Sure.
Yeah, the number of cell types that you had, I think you say you're 30 or so. How did you determine those cell types with the clusters like validated? They're actually different cell types.
Yeah. The question is, how do you know there's 39 cell types? How do you know that each of those cell types is real? How do you determine them? How do you make sense of any of that? So I mean, as far as possible, we're doing unsupervised clustering on the transcriptional states of these cells, mostly using like community detection via Leiden algorithm at many different resolutions, which is essential, right? Because you can do the major cell types, but that just only gets you so far. And the point is, when you have this major variation, then you're not going to be able to see variation between subtle differences in macrophage subsets. So we look at many different resolutions, and at each one, we just look for evidence that there are differentially expressed genes which show a real a transcriptional difference in cell state, which is interesting, which is above the noise and which is somehow reflective of an immune phenotype, right? There's always differences, but is that difference metabolic, or is it some difference in some cytokine signaling? But there's no, I can't give you, this is not automated. Like the algorithm, the clustering algorithms are, but the choice, like all the choices of resolution are very much user oriented and somewhat painstaking.
So my first question is about the I noticed that the agency for a lot of is that really?
Sorry, the question was why are why are other algorithms doing worse on larger sample sizes for classification? I mean, I think that's, I mean, so that comparison, right, is based on simulated data. So it's always really hard, very hard. And like, simulating gene expressions do a good job of accurately like reflecting sources of biological variability. So I imagine what's happening there is that there's noisiness to this data, as well as like true differences. And so the more cells you have, then the more like noisiness you have. So in the case that the true differences are small, then you can start to struggle more to classify a couple thousand cells than to classify a smaller number. So I think that can like lower your AUC as you have more cells. It's all kind of like noise to accuracy ratio. Yeah.
Second question is how I assume that there's no experiment. So have you ever in your model how
Yeah, that's a really nice question. You're right. There's not really any data, and we'd like to measure it. Of course, it would require. The problem with this spontaneous model is you don't know when you're going to get a met. You don't know when to go in and look. So to catch the time point, like exactly when you have some sort of seeding of a metastasis, and then also how do you quantify that is really challenging. But probably like we're going to try to do that experimentally. And I mean, mathematically, whether it's a good question as to whether like the effects of the delay can be like kind of compensated by, for example, other MDSC interaction parameters, like whether you can like recapitulate that without a delay. That's not something that we've like really investigated, but that's a nice idea.
So I wanted to ask John that question. Instead of making it time dependent, like on a delay, why not make it density dependent? On the density itself, like the human needs to be certain threshold to attract that would get at least you can get rid of the problem that there is a time factor, and so every tumor has its own mechanism that it grows. If it doesn't grow, then it will go signal. If it grows, it will always attack.
Sure.
Like,
I think that's sure. We haven't simulated that model. That's a very reasonable sounding alternative model that one could also investigate. I think it requires, like, the nice thing about the delay is that it de-emphasizes the tumors. The tumor has already influenced the MDSC through other interaction parameters. So I think then you would like certainly like it would be you'd have like a non-identifiable case, because then you have like the MDSC reacting to the tumor via two different parameters.
Yeah, I mean, I get it because it's a metastatic tumor, so you already assume that there is a mechanism setting wrong there because it is already the tumor has been somewhere else. So it's a fair and it's more realistic.
But no, but it's yeah. There's right. There's always many ways in which one could construct the system, and that is a nice alternative. You could do away with the delay and have like a time-dependent tumor MDSC interaction to replace it.
I also had like a very clarification for your network. When you're understanding what type of network, do you actually get you are permuting different combinations, different interactions? Do you get the model weights, or do you get the weights for each arrow and exactly tumor, or how is that?
Yeah.
Does this add zero?
So I went a little bit quickly. Sorry, to repeat the question, how do you do the edge weights of a cell-cell interaction network inform the perturbations or the motifs that you get out? So the weights, which are like interaction strength by this ligand receptor inference, they do not inform the perturbations. Those are just like kind of like random uniform across the network. But then the edge weights, once you have a motif which you think is overrepresented, so you ignore the weights to look at the permutations of edges to get motifs which are overrepresented. But once you have an overrepresented motif, then you ask who's signaling via this pattern, And there you rank the instances of that motif with specific cell types according to the strength of the interactions, the edgeways. That's the idea.
Mine's more of a pipeline crisis work. You might have said that maybe I missed it, where the legislation talks about how interaction. Those pathways that you come up with, are those the normal analysis?
No, the pathways are directly from like the ligand receptor, it's like predicted cell-cell communication. So we're looking at like all interactions in these networks are represented by predicted interactions between subsets of gene expression of like ligands and receptors of pathways. So that's where the pathways come from. There's one question here. Yeah, it's the short answer is yes. The question is, are we other mouse models that are better at reflecting different aspects of cancer in a kind of instance and progression? We do have a collaborator who has like the PY mouse, which has like a much more kind of natural initiation of breast cancer, where you see like all these different epithelial cell states as they're transitioning, and like very different cancer progression and a different TME. So we haven't done it, but that's certainly something that we're kind of a place to apply the model. Yeah.
Thank you very much indeed, Adam. Wonderful.
Great.
Nice to see the interplay between mathematics and real data.
Thank you.
Thank you very much indeed.
Thank you.